Hello, Dr. Cristina. Good.
Hello, hi.
Hi, I'm fine. Good day. Dr. Mikro, we're still waiting. Dr. Mikro?
Yep. Hi.
Yeah.
Cuckoo.
Okay.
Hello.
Everybody
Okay.
on.
Thank you, Chris.
You're welcome.
Yeah.
Okay, Dr. Mikro, it's on time at 4 p.m. Can we start for the
Okay.
session?
I'm
Yeah.
ready. Yeah. Yeah.
Okay, before to Dr. Mikro, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Good afternoon, everyone. Thanks for joining this online session in Zoom meeting platform. Before, for those who don't know me, let me introduce myself briefly. My name is Eko Suprianto. I'm General Program Manager in International Office of Universitas Sultan Ageng Tirtayasa. Today, Dr. Cristina Belda Marin is our speaker. We'll be talking about Bionanocomposite design, characteristic, and potential application. Dr. Cristina Belda Marin is a main speaker of this event, will be moderated by Dr. Mikro Permana Pinem. Dr. Mikro is one of the best lecturer in Faculty of Engineering, especially in
No.
Mechanical Engineering Department of Universitas
Oh,
Sultan
sir.
Ageng. Okay, Dr. Mikro, the time and screen is yours.
Okay. Thank you, Mr. Eko. And I think you can censor what Mr. Eko told Christina. I'm not the best at all. I'm really not the best. And um, Mr. Eko is our boss here, so he's our our chef in on France, c'est ça? Yeah, yeah, yeah. I'm glad to have you today, Christina, for this moment. It's really happy. We can have your time here, your valuable time, of course. And I'm I'm also want to. I think I want to introduce you first. Yeah, I took your CV from LinkedIn. I will
Okay.
read some. Yeah, if you doesn't okay, I will read some. So, ladies and gentlemen, I will present Dr. Christina here. She is it's called okay, biomaterials and chemistry. Okay,
Yes.
it's right. Okay,
Yeah.
she was a graduate from I think it's bachelor, yeah, Universitas de Barcelona. Am I right? Yeah.
Yes.
Okay.
Yes.
And after that, she continued to University Grenoble,
Mhm.
and she took PhD in University de Technologie de Compiègne, and she was graduate December 2022. Yeah.
2020.
Sorry, 2020. Sorry, I'm sorry. Okay,
No worries.
and okay, and she was experienced also visiting PhD student to university. Yeah, in Boston. Yeah, United
Yes.
States, I think.
Yeah.
Okay, and now she working as actor in UTC, French.
Mhm.
You still there, Christina?
Yeah, yeah.
Okay. And yeah, I think this is quite interesting. She also active in AFS intercultural programs, which is works from coordinating and preparing meetings with all students and host families to ensure everything is going good, to give advice to students and families and ensure the students are well integrated. Wow, I just noticed this. It's great, Christina. Okay,
Thank you.
we will have you this for your presentation. We will continue for 30 minutes. I think is it okay? 30 minutes,
Yes.
right? Yeah, and we will continue with discussion.
Okay.
Okay. Do you have any question before we start your presentation, please?
No, I don't think so.
Okay. Okay, maybe this is important. She speak in French, for sure. She speak in Spanish, English, and also Italian, no?
Catalan. Oh,
Catalan. Okay. Okay, the time is yours, Cristina. Yeah,
thank you.
please. Okay.
I'm just going to share my screen. Is it working now?
Yep, perfect.
Great. Okay, so as Mekro said, I'm a Spanish person that has been studying in Spain, in France, and I've been visiting in the United States also. So I really like this international view, and that's something about science that really tires my attention, because you can get in contact to everyone. And I'm so happy to be here today, and I thank you very much, Mekro, for this invitation. And I hope you find my presentation very interesting. So what I'm presenting today is my PhD work, which I've been continuing during this year. And this PhD was done within the Université de Technologie de Compiègne in France, and in collaboration with the Laboratoire des réactivités des surfaces in Sorbonne University. Okay, so the title of this work is Silk Biocomposites, their design, characterization, and their potential applications. Okay. I'm going to divide my presentations first to give you an overview of the state of the art about silk biomaterials and what we can do with them. Then we'll go through what silk bionanocomposites we have designed in our laboratory. Finally, I'll end up with quickly some of the applications that we have shown for those biomaterials. So for the state of the art, I think you all know silk much more than me in your daily life. So silk is, of course, a natural product. It's biocompatible, biodegradable, and has adapted mechanical properties. So silk is a very strong material that it's produced from many different animals. The most well-known are spiders and silkworms. So spider silk is much more interesting for engineering because of their mechanical properties are stronger. However, their production in industrial quantities is limited because spiders eat each other, and their silk production is much lesser than those of silkworms. So here we are using the silk coming from silk cocoons, as you see in the background, of silkworms. So this silk, it's composed by two proteins, sericin and fibroin. And fibroin are composed of fibrils, and sericin is coming to surround this fibroin. So some studies have shown that sericin is responsible of immunogenic responses when we are using it as a biomaterial for implantable materials, for example. So much of the studies have been focused only in the fibroin part of silk. So from now on, when I will say silk, I'm actually referring to silk fibroin. So silk fibroin structure is divided in two chains, a light chain and a heavy chain. And the heavy chain, as you can see from the picture in yellow, it has many repetitive domains, which allows the formation of beta sheets, and this rises to crystalline regions, which are the responsible of the so strong mechanical properties of silk. So if we talk now about silk-based materials, I'm sure you all know the silk clothes that we can use and that are derived from the textile industry. So in this, to produce these materials, we have a deguming process, which slightly consists on removing sericin and just getting silk fibrin to form silk threads. If we think about the application of silk in the biomedical field, you will maybe think about silk sutures, which are already used in the medical field. However, there are many more materials that can be obtained from silk and that are less extended, but are very great into the research area. So in fact, the idea is to get silk from silk cones, in which, as I told you, it's structured in beta sheets. We're going to denature this silk to obtain a silk fibrin dispersion. And then by controlling the reformation of beta sheets within the silk, we're going to control the material that we are obtaining. So from this procedure, we have actually many materials in our laboratories, such as sponges, electrospin mats, hydrogels, aerogels, microspheres, films, or even 3D printing. So these are all materials that have been derived from this silk fibrin solution. So as you can quickly see, silk is a great versatile material, as they can be shaped in many different ways. And because of this, it can be used in many fields, such as biomedical, but also as in the pollution fields, as catalyzers, in electronics, or many others. And because we are able to control the formation of beta sheet content, we are able to control through this step the biodegradability of the material. So we can think to have a material that's either biodegrading within 1 week or within 3 months in a tunable manner. Also, the size of the extracted fibrin will be important in this step. So here the idea was to take silk biomaterials that already exist and functionalize them with nanoparticles to give them extra properties. So nanoparticles, as you may all know, or nano objects, are objects within at least one of their 3 dimensions. It's lower than 100 nanometers. So these nanoparticles are used in many fields, such as medicine, electronic, and catalysis, but in daily life, you can find it in paints, a washing machine, or electronics. So they're pretty much everywhere. And what's important about nanoparticles is that their properties are differing from a bulk material. So for instance, gold, a piece of gold will be golden, while gold nanoparticles are not golden. We'll see this by the following. So these properties, are dependent on their high surface to volume ratio, and so dependent on the size of the nanoparticles. Their shape is very important as well, and their surface chemistry. So here, the idea is to produce nanoparticles with a controlled procedure that allows us to control these 3 so important parameters, and then to master the nanoparticles' properties. So this is what's silk bionanocomposites. So bionanocomposites are silk materials that contain nanoparticles within it. So the idea here is like preparing a dish of pasta. We have different types of pasta, which we can add either meat or tomato or cream or whatever. We have bolognese or carbonara, for example. So the idea is to have different materials coming from silk fibrin, in which we are going to add different type of nanoparticles to obtain different bionanocomposites. So the end here were then to synthesize nanoparticles in a controlled manner to master their properties, to synthesize different bionanocomposites that in a manner that can be transposable to different materials and different types of nanoparticles, and finally to characterize the influence of nanoparticles over the silk structure and show that even when nanoparticles are included into silk materials, their properties are maintained. So how are we going to incorporate these nanoparticles into silk? the literature has mainly used these two techniques, which are in situ synthesis or the synthesis of nanoparticles upstream and then the incorporation into silk. So in situ synthesis consists in synthesizing nanoparticles directly within the silk solution or the silk material. So we are going to add metal ions in our case, and then nanoparticles are formed. So this is a very interesting procedure because it takes place in one step. However, silk solution is very delicate, and the incorporation of ions into the solution can induce earth gelification. Moreover, if metal ions are remaining in the material and that we wanted to use for biomedical applications, for example, this could induce a toxic response. And finally, and much more important, as I told you before, properties of the nanoparticles are dependent on the size, shape, and surface chemistry. And this is not a parameter that we can control through this technique. So the second technique presented here is the synthesis upstream of the nanoparticles, and then the addition either into the silk dispersion or into the silk material. So this step allows the control over nanoparticle properties because we have a control over the size and shape by synthesizing them before. However, several steps are required. We still have a possible solidification when adding metal nanoparticles into silk dispersion. And whether we add metal nanoparticles over the already prepared materials, a release of these nanoparticles could be possible. So here, the procedure that we have chosen to utilize is that one, because it allows us to control the nanoparticle properties, which is very important for us, and then to shape it into different materials, reducing the release of nanoparticles. So we have now the overall view. We're going to explain how silk is extracted. So quickly we get our silk cocoons, in which I remember silk is structured in beta sheets. Then we cut them, we boil them, and we obtain this sort of mesh. It's then suspended into lithium bromide and then dialyzed to obtain finally our silk fibrin dispersion. So this procedure has been FDA approved. So it's something that's very standard and already well known. So now the idea is to incorporate nanoparticles into this solution. So first we have to synthesize nanoparticles. Here we want to have methyl nanoparticles that are synthesized in a green manner and not in organic solvents. So the idea is to use hydroxymethylene biphosphonic acid, which are molecules that are already used in osteoporosis treatment, to stabilize those molecules. So the literature has used some of those molecules to stabilize nanoparticles. Here we decided to choose this one because the presence of a triple bond at the end allows a easy functionalization by click chemistry if we want further to add either fluorochromes or bioactive molecules, for example. So the idea is to adapt the syntheses that we have developed in the laboratory for palladium nanoparticles to gold and silver nanoparticles. And what's important about this synthesis is that it's done in one only step within water as a solvent and using sodium ascorbate as a reductant for those metals. The third type of nanoparticle that we have chosen to use within this study is iron oxide nanoparticles, which were synthesized by alkaline co-precipitation, which is the standard manner, and then functionalized with our molecule into the surface. So these three types of nanoparticles, gold, silver, and iron oxide, are already used in the biomedical field, and that's why we have chosen them. So for for gold nanoparticles, sorry, the idea was to take gold ions, place them in presence of our stabilizing agent and our reductant, then 30 minutes at room temperature under agitation allows the formation of gold nanoparticles. And we have studied these nanoparticles by transmission electron microscopy. You see a diameter of 4.7. More important, we have also shown by spectrophotometry that we have the well-known absorbance around 520 nanometers. That's known for gold nanoparticles. So slightly the same procedure was used to synthesize silver nanoparticles, in which we have silver nitride as an ion source. We add our stabilization agent or reductant agent, and this time we need to heat the suspension with microwave during 15 minutes to get our silver nanoparticles. So again, we have spherical nanoparticles that are monodispersed in their synthesis. This time are a little bit larger and without aggregation seen in the dispersion. And again, the light absorption properties are well in accordance to what's seen in the literature for silver nanoparticles. Finally, for iron oxide nanoparticles, we synthesized them by the co-precipitation method in presence of an alkali. Then they obtained iron oxide nanoparticles, were coated with our stabilizing agent, and then placed into a water solution. So as you can see from the transmission electron microscopy image, those nanoparticles tend to aggregate much more than the other two, and that's easily explained because of their magnetic attraction. But still, we have rounded nanoparticles with a monodispersed size. And what's important about iron oxide nanoparticles is that they have this super paramagnetic behavior that allows, for example, their use for magnetic resonance imaging, as we will see later. So now we have nanoparticles, we have silk. It's time to get both together. And in fact, silk fibroin, it's autofluorescence, and this autofluorescence changes along the protein structure. So we have studied the fluorescence of silk solutions. I'm sorry, I just realized that's in French. Yeah. So this is the fluorescence within the wavelength. And, we have studied this fluorescence of the nanoparticles within the solutions of silk, and we have a slightly change for silver nanoparticles, but not for the others. So the change in intensity, I will explain later why we have a lesser intensity. So when adding nanoparticles into silk solution, we have a solution that's still liquid, so no gelification of the silk has been observed. As I told you, the properties of light absorption are very dependent of the nanoparticle size and shape. And so if an aggregation occurs when putting them into silk solution, we would see this through a change of the maximum absorbance. And as you see here for silver and for gold, it's not the case. So we still have the same maximum absorbance for the both type of nanoparticles when they are included within silk solution. So I'm going to explain now something about the silk hydrogels, the materials that we have been focused in. So silk hydrogels can be produced by enzymatic cross-linking methods in which we are actually using horseradish peroxidase to induce a cross-linking of the tyrosines found within the silk structure. And these tyrosines form a deteriorating bond, which are then fluorescence. So this allows follow-up of this hydrogel formation through this the apparition of this fluorescence peak. And here you can see what the different hydrogels containing nanoparticles look like. So transparent is silk hydrogel alone, and then you have iron oxide, gold, and silver. And as you can see, they are very quite strong, as you can push them and they are not break. So we checked whether the fluorescence follow-up was possible in presence of the nanoparticles, and we were surprised because it is possible, but we have a lower intensity. So this lower intensity, it's called a quenching effect that's caused by the presence of nanoparticles, which is explained, for example, for silver nanoparticles. I remind you that the solution of silver nanoparticles absorb light at 430 nanometers. That explains, it's very close to the peak of fluorescence of deuterazine, so it explains this reduction of intensity. Similar to gold nanoparticles, which they have a maximum at 520, but they had absorbance over the whole spectrum. And lower effect is seen for iron oxide nanoparticles, as the absorption of light is much lower than the other two. So we have studied the structure through light confocal fluorescence microscopy and scanning electron microscopy to see that there are not many changes within the structure, and that we actually have similar structure for all materials. And finally, we have seen that all nanoparticles are actually within the hydrogel by two different techniques. So I'm just going to explain some of the applications that we have shown for these hydrogels for the moment. So the first one is for silk containing silver nanoparticles. So the main application of silver nanoparticles are, of course, antibacterial properties. And here we want to characterize whether our silk hydrogel has antibacterial properties or not. So what we do is place bacteria into a petri dish, place our hydrogel containing silver nanoparticles over the bacteria, and just to see after 24 hours whether bacteria are able to grow or not. So here you have the results for E. coli, in which you see that for silk hydrogels, bacteria are happily growing next to the hydrogel. However, for silk silver nanoparticles, a surrounding area of inhibition is seen, and bacteria are not able to grow. We have unexpected antibacterial action. We wanted to know whether this antibacterial action was due to a release of silver ions, as it's explained in the literature for most of the case. So here we put our hydrogel into an agar petri dish, this time without bacteria, to allow the possible release of material. And then we remove the hydrogel and put bacteria over the same petri dish and evaluate whether bacteria are able to grow or not. And surprisingly, bacteria were able to grow in both cases, determining that no or not enough silver or silver nanoparticle are released to avoid bacterial growth. Of course, these materials were tested to be whether they are compatible, biocompatible or not. And our cytotoxicity test, as described in the norms, showed that no cytotoxicity effect was observed. And moreover, human dermal fibroblasts were able to grow over the hydrogel in both conditions. I'm going to pass this step for magnetical applications, and I'm just going through the last application concerning gold nanoparticles, because I think it's more interesting for you. So the third application, so the application concerning gold nanoparticles was for water depollution. So it has been shown in the literature that silk materials are able to absorb organic molecules from the environment. So here we have chosen to use a methylene blue as a model component, which is used into textile industry, paints, everything that's blue has 90% probability methylene blue on it. So jeans, t-shirts, whatever. So here, firstly, we want to study the absorption capacity of those silk hydrogels. And to do this, we are going to get our silk hydrogel, place it into a methylene blue solution. And over the time, we're going to see that actually the entire methylene blue is absorbed within the silk hydrogel. Then we want to combine this absorption capacity with the catalytic properties of gold nanoparticles. Here, the idea is slightly the same. This time we are using our silk gold nanoparticle Bionanocomposite, introducing them into methylene blue, and then over the time, methylene blue will be degraded. So let's start with the first, so the characterization of the silk hydrogel's absorption capacity. So as you can see from the image, when introducing our hydrogel into a methylene blue solution, over the time, the quantity of methylene blue absorbed by the hydrogel increases over time until the time where no more methylene blue is contained within the solution. So as you can see here at the beginning, the hydrogel is in the solution, but we cannot see it. At the end, it is blue because it has absorbed all the colorant. So now we want to combine this property of absorption with the catalysis of gold nanoparticles. So methylene blue, I didn't say, it's a model molecule that we have chosen, but it's very used in the industry and it's toxic and it's pretty much released into the ocean. So it's a major problem. So it has been shown in the literature that gold nanoparticles can catalyze the reduction by borohydride the sodium, and it catalyzes its reduction to leucomethylene blue, which is not toxic and not colored. So it's very good for our experiments. So here we have our cycle hydrogel containing gold nanoparticles, and so you can see from the video, with the time, our colorant is catalyzed. And at the end, we have no more methylene blue, neither within the silk hydrogel, neither within the solution. And very important, of course, this is not the case when we use only silk hydrogel without gold nanoparticles. So we have characterized this through the light absorption of methylene blue, and maximum absorption is found at 664 nanometers. As you can see, within only 15 minutes, we arrive to a zero absorption. So the entire methylene blue has been reduced. Of course, it's important to notice that if you remember, I told you that gold nanoparticles have a maximum absorbance at 520, and here it's not seen. So we can say that there is no release of nanoparticles into the solution, which is very important for us as well for toxicity issues that may appear. So within 15 minutes, our hydrogel is able to reduce the entire solution of methylene blue. Now the question was whether this procedure can be repeated. Of course, the hydrogel can be reutilized into a new methylene blue solution, and we have done this at least over 15 times, obtaining the same results. So the material can be reused and recycled without a problem. So I'm going to finish with conclusions and perspectives. Just saying that in this presentation, I have shown the compatibility of silk fibrin and or nanoparticles, which did not induce the gelation of silk and were able to form hydrogels in presence of nanoparticles. We have not seen a change in this hydrogel structure when nanoparticles were incorporated. Moreover, the properties of such nanoparticles are maintained when they are into silk hydrogels, and we have not seen nanoparticle release in the environment, which is very important concerning nanoparticles today. Finally, I have shown the application of bionanocomposites for silk with silver. We have seen an antibacterial action by contact rather than by release. For iron oxide, I have not shown, but it's possible to do magnetic resonance imaging with it. And then for silver, gold nanoparticles, we have combined action of the silk materials which are going to absorb and the degradation through catalysis for organic molecules. So finally, I just want to say that this method has been transposed to other silk materials such as electrospun materials, 3D printing through different techniques thanks to my time in Boston with Tufts University, and then silk sponge that contain also these nanoparticles. So again, the versatility and adaptability of those materials allows to use them for different applications. So for example, a silver or silver iron oxide mix could be used for wound closure and cicatrization. Iron oxide can be used for MRI, as I told you. And finally, for the depollution, we can imagine to use different catalysts, such as different nanoparticles, include different enzymes into the hydrogel, or combine both enzymes and nanoparticles to have hybrid catalysis. So I'm going to finish my presentation just by thanking everyone that has been working with me and has made possible all this work. So of course, everyone from UTC and LRS, which are the two laboratories in which I've been working, but also our collaboration with LVTS and Neurospin for the iron oxide nanoparticles, and finally Tufts University for all the silk 3D printing and related materials. And I will also want to finish thanking again Macro and your university for inviting me today to this meeting. And I will happy to take any questions that you may have.
Okay, Cristina, sorry, I have a little problem with my device, but I think I can solve it for the moment. Thank you for your presentation. Great presentation for sure, as always. Okay, we have one question here from Gali. Compared to the well-known nanoparticle or nanotubes, TI or the en France, what is the advantage of the 6 nanoparticles? That's the first one. Maybe you can, you can answer directly.
So here the idea is not to use. I don't know if he's talking about an application in a specific application, but the idea here is not to prepare silk nanoparticles, but methyl nanoparticles that are included within silk material. And as I explained, what this can limit, it's, for instance, for the catalysis application, when using nanoparticles, they are dissolved in the media and hard to recycle for another application, as when they are included in a material, they can be recycled, easily recovered. And nowadays, you may all be informed that when you talk about nanoparticles and especially when you say to someone, I'm going to place nanoparticles in your body, the first reaction is like, hell no. Okay, so it's something that's very scary for people, even if I've shown that they are not cytotoxic, they have not toxical results. However, it's something that scares people. So for us, using a silk material, it allows us to still have the nanoparticle application. So either super paramagnetic properties or antibacterial applications, but not to freely release all the nanoparticles within the body or within the water for the pollution or within the system in which we want to use it. I don't know if I did.
Yeah. Thank you for your answer. And we have another one. I think it's good one. The question is: can silk nanoparticles used to reduce heavy metals like PB for the pollution?
Yeah. So silk absorption has been studied for organic molecules, but also for heavy metals, as you say. So there are many studies for PB reduction absorption, also cadmium absorption, for example. And of course, this can be used. And the same example that I have shown here. So silk by itself will be able to absorb those heavy metals, and you can add to it a different component that will able to reduce them or make them lesser toxic by coupling to other molecules, for example.
Okay. Another one from Bintang Syah. This research requires a very high accuracy. How do you deal with everything that could affect this research so that it does not change? It requires talented hand, yeah? No? Okay.
So, of course, for example, silk solution, I said it's very delicate. And so it has to be, whether you use it or not, after a month, it's finished. So it has to be taken care of. And it has to be kept in the fridge. And I usually do my silk extraction right before I want to use it. And then for nanoparticles, I had, to be honest, it was difficult to set up the synthesis of silver nanoparticles, for example, because they are stable. And then here we are talking about a synthesis in water. And then the control over the properties, size and aggregation is very important, of course, for their properties. And so it's very important to do everything in the same manner every time. So that's why everything has been written down, this temperature, whether it was raining or not this day. Like every small change could be, of course, change this. So it's very important to take into account every small parameter.
Okay. Thank you, and we will continue for the next question. And this nanoparticle, what kind of, can you imagine in daily life? What is the application? I think it's question, and how much it can reduce the pollution in percentage.
Sorry, can you repeat the question, Macron, please?
How much it can reduce the pollution in percentage? I'm not sure, for example, in 100 milliliter.
So this is dependent of the time, of course.
Okay.
So if you want to do a reduction,
or it will be like asymptotic, and you will remain always remain something there. No?
So because silk hydrogel is absorbing the molecule, whether if not reduction is done, even the silk, the methylene blue will be absorbed by the silk. So either way, here you have the combination of both the reduction and the absorption of the material. So we have not been able to get to the end of this of the application because we have repeated the step for 15 times and it still works as well. So I want to say that the limiting factor will be silk degradation and not the material that's not working anymore.
Okay. Can silk be the next question? I think, Cristina.
For you, the next question.
Can silk bionanocomposite? Okay. Bionanocomposite be used for food packaging? What do you think of it?
Yeah, so silk have some films have been done with silk. The problem here is that silk, as I told you, is a protein, so very good for bacteria. So when you want to use it into fruits, for example, it's been possible to use it, but we have to very much control whether bacteria will be able to grow within this protein or not. So very, very important to take, for example, the bacteria addition to the material, whether if we have a very crystalline material, bacteria will not be likely to quickly degrade this material. So their growth within the material will be slower, but it's an important thing to control and to take into account from the beginning. I don't know, Macro, if you want to add something as you have work.
No. You are an expert in this. I'm only beginner. Okay. I have. Okay. I want to going further for when you told that when you make like nanoparticle and told to people to it is okay for your body, but people like they hesitant to take it. So in your opinion, in the future, what kind of is there something that it is necessary to have nanoparticle in our body and we have no other choice, or nanoparticle will always like, yeah, it's good, but not for my body? Okay.
Yeah, okay. So let's just take, for instance, the silver nanoparticles, which are today really used for antibacterial actions. And this is very important. And why we are using nanoparticles, it's because silver ions, themselves, they are toxic, but not silver nanoparticles. So very important, silver nanoparticles are lesser toxic than silver ions themselves. And because of the antibiotic resistance that has been developed worldwide, we are now looking for other solutions from, for example, silver nanoparticles. Of course, each type of nanoparticle has to be tested for their cytotoxicity. The choice, because today, for instance, many antibiotics are not efficient anymore, so we have to find several solutions. In the case of titanium oxide that was mentioned before, it's the only solution that we have found for UV radiation protection. So it's what's contained within every sunscreen that you use.
Okay.
So it's, for some applications, we do still have other solutions, but we are arriving now to some limit points into which nanoparticles are really advantageous in comparison to what we are using, such as antibiotics, for example, or UV radiation protection. And so I really think that this will be like a COVID vaccine, for example, in which no one wants to put it, but we have to.
Yeah. That's why I'm talking to. I think it's a point. Yeah. We all maybe in the public like here, especially in Indonesia case, for in my country, we already have nanoparticles around, and we didn't notice it. I think it's a good point, yeah. And
yeah. So it's very important to notice that Nanoparticles are everywhere. So
yeah, nanoparticles
most of
are
the time
everywhere.
we don't know, but they are really everywhere.
Yeah, okay, that's a good point. I mean that if this idea is not popular in my country, especially nanoparticle, and we have no regulation for it, and we just take it for granted and yeah, do as you want. I think it's a good point to make more criticize what industry put it in your food, in your tire, and everything. So I just wondering in terms, especially for your application here, Cristina, if we develop further, uh, because you play in 100 milliliter, for example, to reduce heavy metal or another metal. But how if we work really in industrial scale, for example, 100 meter cube to
Yeah,
cleaning
so
something like
the idea was so actually today, to remove these heavy metals or aromatic components, what's done in industry, it's absorption. So we're going to use a generally activated carbon that's going to absorb whatever material we want to remove, and then the material contain the absorbed molecule. It's going to be burned for the most of the time. So what really doesn't help the problem that much, because we are removing a molecule to finally turn it into carbon dioxide or many other things that are produced when burning the material. So here the idea was to use different small hydrogels to increase the surface of exchange. And of course, this can be used all together in a bigger suspension.
Mm-hmm.
And what's interesting is that this material is going to degrade over time,
Mm-hmm.
and it's biodegradable, so it will not be required to be burned and then continue the pollution. So
Okay. Okay, really insightful. Do we have any question, or you can, for all the audience, you can ask directly to Cristina without writing it in the chat. If you have any, please, you can ask directly, because Cristina is the future scientist. I think she will be really great. So it's really.
So I'm just
really,
seeing in the chat.
uh-huh.
I comment that says silver composite is very less hazardous than its ion form, and I completely agree with it.
Okay.
And for example, it's used in ingredients in soap and its antibacterial activity. Yes, we are now arriving to socks containing silver nanoparticles, t-shirts containing silver nanoparticles. Samsung has even got one washing machine that's using silver nanoparticles because it kills bacteria, okay. And so your clothes are cleaner and smell less. So we are actually using everywhere. And it's very important what you said before about regulation, macro. This is like everything that comes up new. Laws and regulations take a long time to set in place. So at the beginning, we are all lost, like whether it's toxic or not, can we use it or not? But it's the same for medicine, for any material that's discovered.
Yeah, I think it's quite difficult.
And what's important for nanoparticles is that we have to test nanoparticle by nanoparticle. Every synthesis method will change the properties of the nanoparticles. That's what I was saying from the beginning.
Okay.
And this is very important because for silver nanoparticles cannot be taken as a whole, because depending on the size, the shade, they will be toxic or not. They will be adherent to whatever, sorry, they will adhere to your skin or not. So it's very important to take the case per case for toxicity regulations. And it's the same in medicine. So I'm a biologist, so in medicine is the same. Whether you are changing one single atom in a molecule, you have to redo all your tests to see whether your molecule is working or not, it's toxic or not. So everything has to be very regulated, and we are not still there.
Yeah, yeah. What do you mean by we? Europe also as well. You have no regulation for this for the moment?
I think there are some regulations in Europe, but
Okay.
because we don't know what we cannot take all nanoparticles together, so it's difficult to say nanoparticles are banned or not, because some nanoparticles are very good, some nanoparticles are less good. For example, the case of titanium oxide, I take this case because it's something that everyone knows. It's something that it's used for radiation protection, so that's very good. When you go to the beach, you take your sunscreen, great, you don't get burned. However, they are also included into yogurts, cheese, that's very important in France, for instance, because they gave a white color only because of the color. Okay.
Okay.
So here, it's not. I mean, I don't really care to eat a gray yogurt rather than a bright white. Okay. So the problem is that whether you say yes, you're authorized to use nanoparticles, then we are going to find applications that are not really justified for the use of nanoparticles. So it's hard to regulate. There are some regulations in Europe, but not that much for the moment.
Okay. Okay, Christina, I'm really happy to talk with you, and I'm sure all of us here, but we only have 4 minutes left, so I think you can make some summary conclusion. Maybe you can focus on in the future. I mean that all industry will go there because, of course, economic value, they will go there, and we are not ready as a consumer. So yeah, I think I'm happy to hear what your opinion about that.
Okay, so of course, yeah, the economical value is something I didn't talk about. Using reduces drastically the quantity of material that you're using. So for the same application, what I was saying, for instance, the white color of yogurts, if I use titanium itself, I need much more quantity than by using titanium oxide nanoparticles. Okay, so of course, all industry will rather use nanoparticles than the material itself, because the quantity is much more smaller that they need to use. So I think that the important thing to remember is that nanoparticles toxicity has been to studied one per one on the case by case and not taken as a whole. We cannot say that all titanium oxide nanoparticles are toxic or all silver nanoparticles are toxic, but we have to study what's in the surface, what's the size, what's their aggregation state. And this is very important. It's something that people don't have in mind, and they just say like every vaccine is toxic, and it's not the case. It's very important to study one per one and not to take all nanoparticles as a whole.
Okay, we really appreciate your valuable time, Christina, and your knowledge to share with us here. And yeah, merci beaucoup.
Thank you very much for your. Thank you very much for your invitation.
We say terima kasih in bahasa Indonesia, as in case you know, and we are really happy to terima kasih.
Terima kasih.
Yeah, it's good. Your pronunciation is great, and we really happy also if you want to visit Indonesia for sure. Please come here. This is beautiful country for sure.
I will for
maybe
sure.
Maybe in another occasion. Yeah.
Yes.
Okay. Okay.
Thank you very much.
Closing, I think. Okay. I think we have 2 minutes or 1 minute left. Thank you for International Office Untirta, who already organized this event, and we really appreciate that, especially for sharing the knowledge, the important knowledge that we will encounter in future, and I think we need to sounding this important subject to regulators if we can, because we are academic and we can do nothing for them, but at least we can write something and make them notice. So we are not like, oh, what is this? Not like surprising what we will come and we will encounter in future. Okay, and yeah, again, I will say thank you to you, Christina, and maybe we will invite you for another research talk if you have any update about your research. Yeah, I'm sure you will be found something meaningful. So yeah, we're really happy to have you again. So okay,
Right.
okay, Mr. Eko, do we have or maybe Mr. Udi?
Mr. Udi, maybe. Okay. Dr. Cristina Belda Marin, so many big thanks for your time today. Dr. Micro, you're amazing. Ladies and gentlemen, let us give applause to our speakers. That's the lecture from Dr. Cristina Belda Marin.
Thank you,
Our
Chris.
moderator, Dr. Micro Pinam, you are amazing. Thank you, Dr. Micro. Thank you for your very good lecture. May all of us get the value and benefit from this session. Finally, we come to the end of this event. I'm sorry if there are any mistakes. To close this event, let's reciting Alhamdulillah. Alhamdulillah. Amin. Happy waiting for your iftar, everyone. Well, ladies and gentlemen, thank you for your joining and your nice attention to this online session meeting. Thank you, Dr. Cristina, Dr. Mikro. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. See you, Christina. See ya.
Thank you.
Say hi to everyone.
See you, Cristina. See you, Mikro. Thank you.
Bye. Yo.
Micro, are you still there?
you.